Welcome to Transcriptomics Unlocked, where we'll decode the molecular blueprint that drives cellular behavior. In this talk, we'll explore how measuring RNA gives a dynamic view of gene activity, the technologies that make it possible, and how these insights translate into biology, diagnostics, and therapeutics. Let's dive into how transcripts reveal cell state and function in real time. Transcription sits at the heart of cellular dynamics, acting as the first regulated step that translates DNA into proteins, while integrating genetic, epigenetic, and environmental inputs. Because RNA can turn over quickly, it captures rapid adaptive responses that static genome or proteome snapshots miss. In short, transcriptomics provides a real-time window into gene activity and cellular state. This slide walks through a massively parallel sequencing workflow, starting with reverse transcription of RNA into cDNA, adapter ligation, and cluster amplification, followed by sequencing by synthesis that generates millions of short reads. Depending on strategy, single-end reads offer speed and cost benefits, while paired-end reads improve alignment and structural detection, and the resulting digital counts enable robust differential expression analysis. Long-read platforms like PacBio and Nanopore let us generate full-length transcript reads, enabling direct identification of splice isoforms, fusion genes, and allele-specific expression without extensive assembly. While long reads offer higher resolution at lower throughput, they complement short-read quantification by resolving complex transcripts and validating structures that short reads alone may miss, improving isoform-level insights. Single-cell transcriptomics using microfluidic encapsulation packages. Individual cells with barcoded beads into droplets, enabling reverse transcription in a controlled microreaction, then pooling for library prep and sequencing. After data deconvolution, each cell's transcriptome is recovered, allowing parallel profiling of thousands of cells and revealing cellular heterogeneity within tissues that bulk assays average out. Spatial transcriptomics maps gene expression back onto tissue architecture using slide-based barcodes, in situ sequencing, and fluorescent hybridization approaches. Each method balances trade-offs between resolution, throughput, and sensitivity, and choosing the right tool depends on whether you prioritize single-cell spatial resolution, transcriptomy breadth, or preservation of delicate tissue features for downstream analysis. Processing transcriptomic data begins with careful pre-processing and quality control to trim adapters, remove rRNA reads, and ensure clean input. Splice-aware alignment then maps reads to reference transcriptomes with accurate placement across exon junctions. And finally, quantification summarizes expression as counts or TPMs, providing the reliable foundation needed for trustworthy downstream analysis and interpretation. Key analysis concepts include statistical modeling with count-based distributions to handle overdispersion, empirical shrinkage, and dispersion estimation to stabilize low-count genes, and multiple testing correction, like FDR, to control false positives. Equally important is sound experimental design with adequate biological replicates, randomization, and blocking to ensure reproducibility and statistical power for detecting real biological effects. Translating transcriptomics clinically involves several use cases, comparing tumor versus normal profiles to identify upregulated oncogenes, assessing immune infiltration signatures to predict immunotherapy response, detecting actionable fusion transcripts for targeted therapies, and using circulating tumor RNA for non-invasive monitoring. Clinical adoption depends on standardized protocols, robust pipelines, and regulatory validation to ensure reliable implementation. In drug discovery, transcriptomics accelerates target identification and mechanism of action studies through connectivity mapping that links drug-induced signatures to disease states, uncovers off-target effects early, and guides pathway enrichment analyses. Integrating transcriptomic assays into preclinical safety and repurposing workflows helps prioritize candidates and speeds an evidence-based, more efficient development pipeline. Epitranscriptomics focuses on RNA modifications like M6A, pseudouridine, and M5C, detected via direct RNA sequencing or antibody-based enrichment followed by mapping. These marks influence splicing, RNA stability, and translation, and emerging evidence ties them to developmental programs, stress responses, and disease pathogenesis, highlighting an extra regulatory layer that shapes RNA function beyond sequence alone. Combining transcriptomics with machine learning and programmable engineering enables predictive and synthetic control of gene expression. ML models forecast expression from sequence. Generative algorithms design regulatory elements. And CRISPR-I provide functional validation and modulation. Together, these tools support precise cellular reprogramming and therapeutic strategies that leverage transcriptomy-level control for future interventions.